Hello, everyone. I'm Soko, and today I'm going to tell you about the thesis project I've been doing at ILVA in Belgium under the supervision of Sophie Derick. So my project basically consisted on working with the MinION sequencer and seafood, and why. First I will start mentioning that seafood is part of the human diet, and its demand is increasing constantly, and so mislabeling practices. For example, The Guardian a few months ago recently published a short review where at least 44 papers related with seafood fraud were analyzed. And it concluded that seafood mislabelling is still happening in a high percentage. And we can understand mislabeling as a total or a partial substitution of one species for another one, usually one with a lower value in the market. And of course, this can have consequences in health, in consumer confidence, and consumer principles. But luckily, nowadays, authenticity testing methods have evolved, and DNA-based methods are now a good standard for the genetic assessment of seafood. And this is known as DNA barcoding, which consists of the use of standard barcodes in combination with Sanger sequencing. And then this result is compared against a reference database. And while the essence of this barcoding relies on the barcoding gap, which is the difference between sequences from different species. However, the main limitation of this barcoding is that it cannot identify food products that contain multiple species. So to overcome this, an approach that has been recently used is DNA metabarcoding, which combine the barcoding principles but with high throughput sequencing equipment. And well, this method depends on the marker choice and sequencing depth. But of course, as everything, it has also limitations, and this time related with the high cost and the amount of time when using certain equipments. And that's when the MinION sequencer can be advantageous. So this sequencer allows to sequence and obtain data in real time. It is portable, as is the size of a smartphone, and especially it is affordable. So the flow cell, which is equipment where you can put your data, can be reused several times. And so that's why the aim of this thesis project was to assess the feasibility of the MinION for authenticity testing. And this was done in two steps. So the first step was sequencing single species that have been already Sanger sequenced. And then by performing the sequence of mixed samples with no composition to evaluate if with the MinION we can recover all the species, even if there are in abundant composition and are closely related and distantly related mixtures. So starting with the single species, the DNA from 21 commercial species was extracted, and after a PCR to amplify the CO1 and cytochrome B genes. For cytochrome B, 15 out of the 21 species amplify, and for CO1, 70 out of the 21 species amplify. So for these markers, this species proceed with the sequencing process. And well, for this single species and as well for the mixed samples, the genes were amplified, CO1 and cytochrome B, and then 3 libraries were prepared for MinION sequencing. And that means that 3 runs were performed. So the first 2 runs contained cytochrome B samples, and they lasted for about an hour and a half. And then the last run contained the CO1 samples, and it lasted for about 18 hours. So then the MinION data was processed with Dicona, which is a bioinformatics pipeline that with a single command allows you to produce consensus sequence ready to compare against a database. So it aligns, it filters, and it clusters the data. And in this case, we compare those sequences against the GenBank nucleotide database. So for the first step of the thesis, the single species, bubble plots were made. So in the y-axis, you can see all the species that amplify with each gene marker. So in the first graph are cytochrome B species, and in the second one, CO1 species. And in the y-axis, there are the species that were found or the species that got reads assigned from the data. Green bubbles represent a good match, so reads were assigned to the correct species or the species we were expecting, and the red bubbles represent a mismatch. And well, the size of the bubbles is related with the amount of reads assigned. So first, for cytochrome B, 9 out of the 15 species were correctly identified, and 3 species were ambiguously identified, which means a certain amount of reads was assigned to another species. And then 3 species were wrongly identified. And well, for CO1, 8 out of the 17 species were correctly identified, while 4 were ambiguously identified, and 5 species were wrongly identified. And well, when trying to find an explanation for these red bubbles, first I will do emphasis on the tuna species. So here, the first 2 tuna species, Tunus orientalis and albacaris, were correctly identified with cytochrome B, while for CO1 were wrongly or ambiguously identified, and vice versa. The correctly identified tuna for CO1 was ambiguously identified for cytochrome B. And well, when analyzing all the data from all species, it turned out that the sequences from the tuna species have no barcoding gap, which means there is an overlap between sequences of different species, and this can mean that it's difficult to discriminate between them. So that's a possible explanation from this group, the tunas. But what happened with the other red bubble species? But well, when tracing back the process and all the lab work, it is possible that a contamination occur, especially during the DNA extraction, and I will mention this later on. But well, from the second part of this thesis, the mixed samples, 2 sets of mixtures were made. So the first set of mixtures was made out of DNA extracts. So 5 nanograms dilutions were made out of the single species extracts, and they were pulled together in different types of mixtures. So my first 2 types of mixtures contain all the cytochrome B and CO1 species that did amplify. So in this heap map, in the first 2 columns, first the gray squares represent the absence of a species, and then in the first column there are the expected cytochrome B species and its reads distribution, and then in the next column there are the results after MinION sequencing. So for cytochrome B, 6 out of the 15 species were retrieved. As for CO1, 10 out of the 17 species were retrieved. And in both cases, there is a different distribution of reads, when it shouldn't be the case, because all species were pulled with the same amounts of DNA. So my third type of mixture was made out of closely related species. In this case, I choose the flatfish group. So for cytochrome B, 2 out of the 6 species were detected. For CO1, 4 out of the 6 species were detected. And as well, there is a different distribution of reads. But the interesting thing is that when combining both marker results, cytochrome B and CO1, then all the species are detected. And well, the fourth type of mixture was made out of distantly related species. So this time for cytochrome B, just 2 out of the 8 species were detected. As for CO1, it was 4 species. Also an interesting thing is that when combining both barcodes information, all fish are detected except for the invertebrate species. And well, then I will go to the second, the last type of mixture I made out of the DNA extracts, and it's a 65 ratio mixture. That means I put a dominant species, 60 nanograms, and then I put 5 nanograms from other 8 species. And well, here the good or the positive thing is that the dominant species got the majority of reads assigned in both cases, but for cytochrome B, just 2 out of the 8 love-abundant species were detected, and for CO1, 4 out of the 8 love-abundant species were detected. And then for the second set of mixtures I prepared, they were done by excise tissue, homogenize it with a pestle, and then proceed to the DNA extraction. So the first type of mixture was made out of distantly related species. So for cytochrome B, just 3 out of the 8 species were detected, while for CO1 it was the same, 3 out of 8 species. The reach distribution is still different, it varies, but such as for the DNA extract mixture, when combining both results of the markers, all fish are detected except for the invertebrates. And then, well, the last type of mixture I made from this set is also a 65 ratio. So I pulled 60 milligrams of Tunus albacaris and then 5 nanograms or other 8 species. And here is the same pattern. The majority of reads were assigned indeed to the dominant species, but in this case, just one lava-abundant species was detected with cytochrome B, and then with CO1, 4 lava-abundant species were detected. And well, in all mixtures, the reads distribution was different as expected. And when trying to find an explanation, for example, for the mixtures that weren't present in the sample, then one option could be due to PCR bias. Even if some species amplify with certain barcodes are not present, what could happen is that the amplification of some species could be inhibited because of the primers. They could have preference between species. And then the second one could be due to sequencing depth, because in all cases, the CO1 species were always more. There were more species detected with CO1, and this could be due because CO1 was run for 18 hours versus the 1 hour and a half that cytochrome B samples were detected. And well, what I conclude out of this set of experiments was what indeed with the metabar coding, the species identification was highly dependent on the marker choice and sequencing depth. And an important thing I thought about is that the limit of detection needs to be determined, and that's especially important when talking about real seafood samples. It is important to set threshold to discriminate whether it's just a contamination or if it's indeed a case of mislabelling. So further experiments with mixtures of different ratios could be done to know whether which is the minimum amount of tissue or DNA the sequencer can detect. And well, overall, with posterior fine-tuning from the DNA extraction to the comparison against databases, this method using the MinION and a metabar coding approach can be a promising tool for the analysis of seafood mixtures. And well, talking about the DNA extraction and contamination, I think an important thing to mention is that negative controls are needed for further experiments. And well, that was my presentation, and thank you so much. And if you have questions, I will happily answer.